my great pleasure to introduce our speaker for today's colloquium. Dr. Daehwan Kim is a postdoctoral research fellow in the Institute of Genetic Medicine at Johns Hopkins University. Prior to that, he received his PhD in computer science in Professor Salzberg's lab, Steven Salzberg's lab at the University of Maryland. His research focuses on developing novel algorithms and high-performance computing approaches for next-generation sequencing analyses, with a focus on efficient sequence alignment and the discovery of transcription gene fusions. Daehwan was the first and corresponding author on several very highly cited papers on the methods he has developed, which became some of the most widely deployed bioinformatics software in use today. So one example is TopHat2. It was published in 2013 and was cited 2,600 times since then, and 1,200 times since 2016. So today he's going to talk about his more recent work on novel ways of representing and efficiently searching a population of human genomes to detect genomic variants from new individuals' genome data, which can facilitate the advancement of personalized medicine.
Thank you very much, Suin, for that kind introduction. And thank you for inviting me to talk about my research. Here, I will describe some of my research projects in computational biology. As technologies have advanced, it will soon become possible for anyone in this room to have your own genome sequenced. In this talk, I will explain some of the key computational challenges in analyzing individual genomes and some innovations I have made to help address them. I've created what's currently the fastest alignment program, HISAT, and extended the program to represent and search a population of human genomes, which enables us to effectively handle sequencing data from any individual. And I've created the first method for assembling HLA genes, which are very important for organ transplantation using whole genome sequencing data. Before going into detail, I just want to go over some points in genomics to set the stage for understanding my research, and I will explain some key concepts and terminology as we go. As we know, the cell is a basic building block of an organism, and it's estimated that an adult human body consists of 20 to 50 trillion cells. Each cell has its own DNA, consisting of 4 types of nucleotides, A, C, G, and T. And DNA is the program code that directs virtually all the events happening in the cell and in an organism. DNA is stored in a double helix structure where A pairs with T and C pairs with G, each together forming a base pair. In the human cell, we have about a total of 6 billion base pairs of DNA. So if we were to store 6 billion base pair or nucleotide DNA sequence in books, we would need thousands of books shown here, and it also needs 1.5 gigabytes of disk space to store the DNA sequence using 2 bits for each nucleotide. So the 6 billion nucleotide DNA is split into chromosomes, chromosomes 1 to 22, and chromosomes X and Y. Typical human cells have pairs of chromosomes from 1 to 22. Females have a pair, 2 X chromosomes, and males have 1 X and 1 Y chromosome. Altogether, these chromosomes constitute our human genome. Human genomes are nearly identical, such that any 2 individuals have approximately 99.9% identical genome sequence, with 1 nucleotide difference every 1,000 nucleotides on average. Genes are segments of DNA that encode proteins, and it's estimated that we have 20,000 to 25,000 genes in our human genome. Differences in our DNA sequences are the main factors that determine our varying phenotypes and are responsible for many diseases. There is a wide array of identified genetic disorders involving point mutations, just the one base differences, or large-scale genomic differences. Only a few of them listed here and many more remain to be discovered. For example, one base difference in chromosome 4 can lead to dwarfism, and having an additional chromosome 21 leads to Down syndrome. So knowing the exact sequence of our DNA is essential for predicting the risk of getting certain diseases and enhancing personalized medicine. So now, I'd like to explain what kinds of resources and tools are available and that enables us to understand our DNA, such as the reference human genome and high-throughput sequencing technologies. Then I'll go over some of the key computational methods to get the most out of these resources and technologies. So you are probably familiar with the Human Genome Project. The project began in 1990 and was completed in 2003. The goal of the project is to construct the reference human genome. And one may think there are already technologies that can read each of these tiny and entangled chromosomes as a single unit. But unfortunately, we did not and still don't have such technologies. So researchers instead had to extract lots of small DNA fragments, read them somehow, and stitch them together to build human reference genome. So it took many years and a lot of money and a lot of people around the globe. So the human reference genome is haploid reference, meaning that it consists of a single set of chromosomes. So the reference genome is 3 billion base pairs long instead of 6 billion. Although today we could assemble our own genomes with the approaches used in the Human Genome Project, it's still very expensive and time-consuming, even with current technologies. So let me give you the basics of DNA sequencing. So first, we extract lots of DNA molecules from many cells, and this could also be RNA as well. Then we chop them up into small fragments. For example, this fragment came from this longer DNA molecule, and we use sequencing machine to read this fragment, shown here, showing CAT, TCT, and so on. And this sequence is called a read, and we can generate many short reads, shown here. We now have high-throughput sequencing technologies, which I often refer to these as next-generation sequencing technologies, which were introduced in mid-2000 and have been dramatically improved over the past decade. So Illumina's HiSeq platforms are the most widely used sequencers, and one of the models, HiSeq 4000, can read in a day 400 billion nucleotides, which is 130 times more than the size of the human reference genome. NovaSeq 6000, which was released earlier this year, can read in a day 3 trillion nucleotides, which is 7.5 times more than HiSeq 4000 platform. So using these machines, we can have a lot of reads that altogether cover the whole human genome, say over 100 times or more. However, these platforms can still only read short DNA fragments, typically 100 to 150 nucleotides long. So the reads may not provide useful information unless we know where they originated. So this is where the human reference genome comes into play. So as I mentioned before, our genomes as humans are nearly identical. So if we can match or map the reads or DNA fragments to the reference human genome using computer algorithms, we could say where a read originated and from which chromosome and which location on the chromosome. For example, a read may map to chromosome 6 beginning at the 2 millionth base. Since reads are from an individual, usually different from those that constitute human genome, the reads are not always exactly the same as the reference genome. Additional differences can also be introduced as errors during the sequencing steps. So I like to describe how reads may have differences with respect to the reference genome. So this is part of chromosome 6 of the human reference genome. And the read number 1 is the same as the reference genome with identical bases shown in green. So this is type of exact matching. Read 2 is nearly identical to the reference with 2 mismatched bases shown in red, T versus C and C versus G. Read 3 has an insertion of 1 base, A, which the reference doesn't have. Read 4 has 1 base deletion with respect to the reference genome. So if we use a brute force alignment algorithm to align to match reads, for example, if we compare a read to the whole genome from beginning to end using some string matching algorithms that allow for mismatches and gaps, alignment can take a very long time. Quantitatively aligning n reads to length of m to reference of length d would take time proportional to n times m times d, or in mathematical notation, big O of nmd. A typical value of n, the number of reads in a given sample, is 2 times 10 to the 9th. M, the read length, is 100 bases long, and d is the size of the reference genome, 3 billion bases long. So NMD is 6 times 10 to the 20, which is a tremendous amount of time. So we may need many years with many CPU cores to align the reads from a single experiment. And there are many computer algorithms available to process reads much faster than this. And I will soon describe some of my methods, which are currently the fastest alignment programs. So my research has primarily focused on developing methods for analyzing next-generation sequencing data. And here I'd like to briefly go over my past and current research. So I first developed a novel algorithm called top-hat fusion to discover fusion events. So chromosomes sometimes break off and join other broken chromosomes. And these genomic translocations can often result in cancer. So top-hat fusion significantly reduced the false positive discovery of fusions, the biggest problem suffered by other fusion finding programs, while allowing for more sensitive detection of the fusions. And top-hat 2 is one of the widely used alignment programs for RNA sequencing data, originally developed by Cole Tremner here at UW. And I led the development of top-hat fusion at top-hat 2, which includes several enhancements. In the meantime, the volume of sequencing data has grown tremendously. So to keep up with this big data, I developed a program, HISAT, which is dramatically faster than other computing methods. HISAT uses an indexing scheme called hierarchical indexing, which employs 2 types of indexes for alignment. And this is what enables the fast alignment, for example, making it 50 times faster than top-hat 2. Our current genomic analysis, including my methods, such as HISAT and top-hat 2, is based on the reference human genome. In order to extend the reference model, I developed a program, HISAT-2, that includes novel methods to represent and search a population of human genomes. And this new way of representing a population of genomes, along with very fast and memory-efficient search capability, allows us to produce much more accurate and detailed variant analysis than previous methods. And recently, I led the development of Centrifuge, which is a novel classification engine that enables rapid and sensitive and accurate labeling of sequencing reads and species abundance analysis. So an environment sample such as soil, seawater, saliva, and so on typically includes thousands of microbial organisms, and Centrifuge sorts out sequencing reads to the species they belong to, for example, E. coli and H. pylori. So first, I will describe how HISAT achieves its fast alignment, then how HISAT-2 aligns sequencing reads to a population of genomes. Then I will go over HISAT-genotype as next generation genomic analysis software. So HISAT was originally developed for RNA sequencing reads, and one of the main applications of RNA sequencing reads is to analyze differential gene expression. So here we have two genes, and the one on the left is transcribed at a higher rate than the other one, and resulting in many more RNA transcripts, and then usually leading to the production of many more proteins than the other one. So the number of RNA sequencing reads we will get is proportional to the number of RNA transcripts here, so which enables us to calculate relative gene expression. And the alignment of RNA sequencing reads is more challenging than the alignment of DNA sequencing reads. As you know, genes consist of exons and introns, and it's the exons that encode proteins. So the cell transcribes gene into pre-messenger RNA, and the cell removes the introns and stitch the remaining exons together into messenger RNA. And we capture this molecule, and we chop it up into small fragments, and we sequence them. And one of the reads is shown here. And when aligning reads to the reference genome, any alignment algorithm should be able to handle the gaps due to intron. So this read spans exon 1 and exon 2, so we need to allow for the gap due to this intron in the alignment process. Computer scientists have developed many data structures, which we call indexes. And these indexes function somewhat similarly to book index, which we may use to find the page that a sentence is on in the book. So there are many indexing schemes, such as K-mer table, suffix array, BWT-FM index, and so on. Each indexing scheme has its own advantages and disadvantages in terms of memory usage and search speed. I will not be going into detail about these indexing schemes today, which will be another hour-long talk. So suffice to say, for the moment, we use the BWT-FM index to search for any sequences of any length that exactly match the reference genome in a rapid and memory-efficient way. So instead of focusing on developing a new way of indexing, I went into a different direction. So I came up with novel indexing scheme, which I call hierarchical indexing, which uses two types of indexes. One is global index that represents the whole human genome, and the other consists of numerous small indexes, which I call local indexes, with each representing a small region of the genome. And there are 48,000 local indexes to cover the whole human genome. For example, the first index represents the first 64,000 bases of chromosome 1. And the next index represents another 64,000 bases that partly overlaps the region covered by the first index. And these overlapping regions make it easier to align reads that will otherwise span two separate genomic regions. One of the main advantages of using hierarchical indexing is that search through local index is much faster than search through global index. Global index is relatively large, about 2 gigabytes for the human genome when represented as FM index. Search through global index generates many cache misses, which adds a lot to runtime, whereas local index is less than 32 kilobytes, small enough to fit into cache, generating fewer cache misses. So, and I will explain the second benefit of using local indexes with the following examples. So this is a genomic region of human chromosome 22, where we have two exons shown in yellow, and their genomic coordinates are shown just below. And suppose this gene is transcribed and the intron is removed, and this is the transcript. And suppose we have two reads shown in red, sequenced from this transcript, and the reads are 100 nucleotides long. One is exonic read, lying within exon 1, and the other spans exon 1 and exon 2, with a short segment on exon 1, 8 nucleotides long, and a long segment on exon 2, 92 nucleotides long. And we will use 3 search operations to align these reads. One is global search, which is based on the global index. Another is local search, based on the local indexes. And extension, the third is extension that directly compares a read sequence with the corresponding genomic sequence, only if we know where to compare. So the extension is the fastest operation which we want to use over the index-based search operations whenever possible. And local search is the next fastest, much faster than global search. So first we'll align this exonic read. And in order to identify candidate locations for this read, the only operation available to use is global search. But the key here, we want to minimize the use of global search. So assuming we align the read from the right to left, and we want to anchor the read to the genome using only portion of the read. For example, using 28 nucleotides of the read from the right allows us to anchor the read to the reference genome uniquely for this particular example. And then we compare the rest of the read with the corresponding genomic sequence with extension operation, which completes the alignment. The next example is in which a read spans 2 exons with a short fragment on exon 1, 8 nucleotides long. So just like in the earlier example, we aligned a portion of the read from the right using the global search, and which also uniquely anchors the read to the genome for this particular example. And then we further extended the alignment, but this time the extension cannot be completed because of this intron and exon boundary, while the read is exclusively an exonic sequence. So we are supposed to compare this short segment with exonic sequence here. So, and if we have only one global index, as is the case in other alignment programs, to search for this short fragment, 8 nucleotides long, we'll end up having a lot of potential mapping locations, because this 8 nucleotide short sequence is everywhere in the 3 billion nucleotide reference genome. So on average at 46,000 locations. So examining all these locations could be time consuming. So some aligners may give up, or others may align it at the expense of much greatly increased runtime. So this is where local indexes come into play. And we know the location up to here, so we can choose one of the local indexes that includes the location and nearby genomic sequences. And when we search for this short fragment, 8 nucleotides long here, using the local index, it's actually mapped uniquely, and which we know is the correct alignment. And we combine these 2 partial mappings to produce the whole alignment of the read. So as you can see, local indexes reduce the number of potential mappings by enabling us to look at nearby regions instead of different chromosomes or on the same chromosome far from this likely location. And using combinations of these operations also allows us to handle mismatches and gaps, which I won't go detail. So based on a simulated 20 million RNA sequencing reads of each read 100 nucleotides long, HISAT aligns the reads at the fastest speed, over 110,000 reads per second, with alignment accuracy at 97.6%, higher than some other widely used programs. I'd like to note that hierarchical indexing can be applicable to other indexing schemes to improve alignment speed and accuracy. So the reference genome, most of our genomic analysis is based on, is rather biased since it was assembled using a few individuals with actually over 70% of the reference genome sequence coming from only one person. So of course, the reference is missing a lot of genomic variants of the human populations. So the alignment protocols are based on the reference genome. They are not often able to align with or produce incorrect alignments, especially when reads from regions of a source genome are quite different from the reference genome. And this can also introduce quite a bit of biases during downstream analysis, such as gene expression analysis, variant calling, screening for genetic disorders, and so on. So in order to address these issues, I developed a program, HISAT-2, to represent and search a population of human genomes. So basically, given the reference genome as a backbone, I incorporated known small variants into the reference genome using a graph. In the near future, I plan to incorporate structural variants, such as long insertions, deletions, and inversions. I also plan to incorporate sequences from other individuals that are not in the reference genome. So I like to illustrate how to incorporate variants into the reference genome using a small example. Here we have 6 base long reference, G, A, G, C, T, and G. In this graph representation, bases are represented as nodes, and their relationships are represented edges. And we have 3 variants, one with a single nucleotide polymorphism, T, instead of second base A, another with 1 base deletion of T between C and G, and we have 1 base insertion between T and G. So briefly speaking, I released the first version of HISAT-2 in September 2015, which requires only 6.7 gig of RAM for representing human genome and 12 million small variants, including 1.3 million insertions and deletions. Because of its low memory requirements, HISAT-2 can be executed on desktop, and the alignment speed is quite fast, comparable to HISAT. So given these capabilities, HISAT-2 is currently the most practical and quickest method available for aligning sequences to a graph representation. HISAT-2 is based on several key technologies, one of which goes back to as early as 1994, namely Burroughs-Wheeler transform. The transform was originally developed for data compression, which basically converts a string to another string, and the latter is more appropriate for high compression. 6 years later, Faragina and Manzini added some metadata to enable fast lookup on this transform while making use of BWT's memory-efficient capability. Bowtie2, developed by Ben Langmead, is one of the most efficient implementations of the BWT and FM index. But these are only for linear string, linear sequence. So recently, Siren et al. Developed an extension of BWT to support graphs. So in HISAT-2, using these methods combined with the hierarchical indexing in HISAT, I designed and implemented memory-efficient indexing scheme called Hierarchical Graph FM Index, which I will explain now with the same example as in the previous slide. So let me provide some basic terminologies. So if you just follow edges, so such as G followed by A, followed by G, followed by C, and this defines a path, and also path defines a string by concatenating the labels of the nodes along the path. And strings can be ordered lexicographically, just like a standard English dictionary. So for example, AGC comes before GTG, which comes before TGZ. And we have a special symbol, Z, to indicate the end of the graph. that is used for properly sorting strings and some other purposes. So far, we may not have a way to search this original form in a memory efficient and fast way. So that's why we convert this graph to the one below called Prefix-sorted Graph. The Prefix-sorted Graph is more appropriate for fast search and efficient storage. But these two graphs are equivalent in the sense that they define the same set of strings. In the Prefix-sorted Graph, nodes are sorted in a way such that any strings from a node with a higher rank comes before lexicographically strings from a node with a lower rank. For example, any strings from the node first ranked comes before any strings from any other nodes. For this prefix-sorted graph, we have an equivalent table where we store two types of information. First, for outgoing edges, given node rankings 1 to 11, we store the label of each node according to the number of outgoing edges it has. So here I'll refer to node rank as node ID. So for example, node rank, node ID 1, has one outgoing edge here. So we store this node's label A once. And node ID 3 has 3 outgoing edges. So we store this node's label C 3 times here. Second, for incoming edges, given the node IDs rankings, node ID 1 has incoming edge from this node labeled G. But this time we store the label of the preceding node, which is G. And node ID 5 has 2 incoming edges from these nodes labeled A and T. So we store A and T accordingly. So although we do not store directly edge information using node IDs, we can easily construct the edge information using a very important property of this prefix-sorted graph called last first mapping property. So last first mapping, LF mapping, is that given last column and first column, the ith occurrence of a certain label in the last column corresponds to the ith occurrence of that label in the first column. For example, the edge between node ID 5 and node ID 3 is implicitly defined using this LF property. So if you look at node ID 3 here, it has an incoming edge from a node labeled G. And notice that this is second G in the last column, so goes to second G in the first column, and then corresponding node ID 5. So this is exactly same as this edge in the graph. And we will use this LF property to search for sequences. So this is original graph where we have 3 base long query shown in blue. And this query involves 1 base insertion with respect to the reference. So we will search this query from right to left. So first G, followed by A, then T. And assuming we know which nodes have G as label, such as 4, 5, 6, 7 in the prefix-sorted graph, and then shown in green in this table. And then when we search for A, we want to look at incoming edges of those nodes. And then we know 2 of them coming from nodes labeled A. So these 2A go here at node IDs 1 and 2 respectively. And then when we search for T, we want to look at incoming edges. So we go around, and then we know one of them is T. So this T goes here at corresponding node ID 8. So this is final alignment in the prefix-sorted graph. And then we have some algorithms to convert this to the corresponding alignment in the original representation. So one thing to note that is that in order to perform LF mapping, we need to know where the ith occurrence of a certain label is, which involves counting the number of label from the top of the table, from somewhere in the table. So this could be time-consuming for large sequence, such as the human genome. And then we can speed up this process by breaking down the whole table into small blocks where we only store a few hundred labels. In addition, we store some additional numbers, such as how many A's have appeared to a particular block, how many C's have appeared, and so on. So instead of counting all the numbers of labels from the top of the table, we just use the numbers stored in a block, and then we just need to count the number of labels within this block. And we optimized this local counting process as well. So my contribution was to design and implement this indexing scheme called GraphFM index. And I also applied hierarchical indexing to this GraphFM index. So the only visible difference is with the one I showed before is that local graph index represents slightly smaller genomic region, 56,000 bases instead of 64,000 bases, because we need room to store genomic variants. So this indexing scheme is called hierarchical GraphFM index, which allows for very fast and memory efficient alignment of reads to graph representation. So this graph-based alignment also significantly expands our ability to read our DNA sequence. So one of my current research projects, HISAT-genotype, is develop a way to look at our DNA beyond a single or few genomic variants identified at the read level. We might be interested in reading whole sequences of genes instead of just a single base difference, such as whole HLA gene sequences or sequences for DNA fingerprinting loci and so on. So we have wet lab-based technologies to do this, but they usually work on one genomic region, say one gene at a time. And now, using the sequencing data from next generation sequencing platforms, I've been developing in parallel HISAT-genotype that will eventually be able to analyze our whole human genome with its over 20,000 genes. That can be done within a few hours on a desktop. So for my preliminary work, I chose one gene family, HLA genes, which are located on human chromosome 6. HLA stands for human leukocyte antigen, which is responsible for our immune defense. These HLA genes are also very important organ transplantation, so it's essential to have a good match of donors and recipients' HLA genes for the success of organ transplant. HLA genes are among the most highly polymorphic human genes, so if my method works for HLA genes, then it would be straightforward to extend my method to analyze other relatively less diverse human genes. So HLA genes have been relatively well studied, and as of now, over 16,000 sequences of HLA genes from many individuals are deposited in the IMGT HLA database. These sequences were identified using different low-throughput technologies. So each person has two versions of an HLA gene, one from the mother and the other from the father, shown in yellow with variants shown in blue. So each of these sequences is called an allele. Alleles may come in different lengths and have many differences with each other. So my goal, given whole genome sequencing data, is to first identify reads that belong to this HLA gene, and then assemble reads into two full-length HLA alleles. So first I designed a graph reference where I incorporated variants of numerous HLA alleles using nodes and edges. I also incorporate variants of other regions of human genome. And whole genome sequencing reads include whole genome sequencing data includes reads data from the whole genome. So we want to extract the reads that belong to the HLA genes using the graph reference with HISAT-2. So based on my experiment, for the read extraction, I got twice as many reads than from using the single reference only. And before moving on to the next stage, let me briefly describe one additional sequencing technique called parallel sequencing. So when we sequence DNA fragments, which are typically 300 to 600 nucleotides long, and current Illumina sequencing technologies allow us to sequence 100 to 150 nucleotides long sequence. So it's common to sequence both ends of the fragments. So this pair of reads called parallel reads are from the same fragment and from the same chromosome and close to each other. So we'll use this pair information when resolving assembly ambiguity, which I will explain now. So instead of directly assembling reads, I split reads into small fixed length fragments called K-mers, which enables us to systematically assemble HLA alleles. For the sake of discussion, let's assume reads are 5 nucleotides long and K is 3. So here we just want to assemble 2 HLA alleles given the read alignments. So for example, this pair of reads are aligned at the 3rd location and 10th location of the graph representation of, for the HLA gene. And this graph shows K-mers from the read alignments. For example, this read consists of 3 K-mers, TCG, CGC, and GCT. So some of the reads have same K-mers, and then some of them have different K-mers, shown here at this junction, branching out 3 different K-mers. And then after these nodes, the K-mers are the same again, and then we have another branch, another junction, shown here branching out 2 different K-mers. So assuming normal cells have just 2 alleles, not 3, not 4, one of these 3 K-mers is likely noise, meaning error, generated during the sequencing or alignment stage. We can basically eliminate one of these 3 K-mers using the number of reads that support each K-mer. So let's say the K-mers in green and yellow are supported by 3 reads each, while the K-mer in red is only supported by 1 read. So the program then simply removes the K-mer in red from the graph. So here we have additional issue, which is assembly ambiguity. For example, we do not know whether CGC, shown in green, is connected to TCG in yellow or CCG in green. So this is where we want to use the pair information. So suppose we have 3 pairs that support the K-mers in green, one of them shown like this one here. And suppose we have 3 pairs that support K-mers in yellow. So using the pair information, now we are able to determine the green K-mers from the same allele, and similarly for these yellow K-mers. So now we have 2 alleles assembled. So I ran HISAT-genotype on real sequencing data sets for 70 individuals with the pedigree shown here. And HISAT-genotype correctly assembled all alleles of one HLA gene, which is HLA-A gene. And although not shown here, I also discovered some novel HLA alleles. So HISAT-genotype is the first method using next-gen sequencing data that successfully assembles some of the HLA genes and enables us to discover novel alleles with high accuracy. So as I demonstrated with HLA genes, HISAT-genotype has the potential to rapidly analyze or 20,000 human genes or so. So in order to perform genome-wide analysis, I plan to provide some template examples that researchers with the right domain knowledge can easily use to put in their own routines. So this way, HISAT-genotype can draw upon many human gene databases and generate output in formats that are relevant to researchers and clinicians. So I'd like to thank Stephen, who is my advisor, for giving me a lot of freedom and flexibility in my research and his support on these projects. Ben is one of my main collaborators. Gio, Lee, and Florian are also my close collaborators. There are also numerous other collaborators who gave me a lot of suggestions and feedback. So that's what I have to share with you today. And I'm happy to answer any questions you may have.
Any questions?
So when you're doing your short variations with the graph, how I see what the model is. How do you actually get the data in there to create those graphs?
So after the Human Genome Project was completed early in 2000, there were many large-scale sequencing projects that generate huge catalogs of genomic variants in the human populations. So one example is DB-SNP database that has over 140 million small variants available in the database. So I have the human reference genome, 3 billion base pairs long, and then so here I incorporate 12 million variants into the reference genome using a graph.
I have a question. So do you use any kind of haplotype information?
I have some implementation. So the thing is, as we incorporate more variants, it actually creates more paths in the graph, and it could be exponential. So as we had more combinations, the index size gets exponentially bigger. So that's actually I use haplotype information. So haplotypes, is simply a set of variants inherit together from a parent. So using the haplotype information, we can actually reduce substantially the exponential combination of variants.
Any more questions? I actually have one more question. So in the HISAT-2 method, you mentioned the incorporation of a DB-SNP and then DB-gap SNPs and so on. Do you incorporate like more dynamic changes?
Like structural
rearrangement,
variants.
yes.
So I plan to incorporate structural variants. First, I like to incorporate long insertions from other individuals. So I expect my genome may have many sequences that are not in the reference genome. So I've been collaborating with my advisor and his PhD student, Rachel Sherman. We recently discovered over 25,000 novel sequences of at least 1,000 bases long from 917 whole genome sequencing data sets from African ancestry individuals. So I plan to incorporate many large insertions to this representation.
Okay. Julia?
So in the HISAT approach, you first align the 28 nucleotide long sequence and then use global local search and the extension. So why do you choose 28? And if the 28 first anchor sequence didn't align to multiple places, then how do you resolve that?
So are you talking about the first example or the second example where?
Like in both cases, you first align a short fragment of the 100 base pair long, read to the genome, and then you extend it or do local search. I'm asking why do you choose 28 instead of longer or shorter, and also when the 28 aligns to multiple places, then how do you resolve that?
That's a great question. So first we need to anchor a read to the genome. So many programs such as bowtie2 use 20 as anchor segment. And then 20, I think it's quite decent length, but sometimes 20 might be really small, and the 20 base long segment can be mapped many locations. So, and then we have many mapping locations. We have to go over those locations to figure out which one is the right one. So that sounds like a time expensive. So I thought maybe I could increase the seed or anchor length a little bit more, and then I can decrease the number of mappings. So that's what I did. So there is some trade-off. So bowtie2 uses 20, so it might be more sensitive, but at the expense of increased runtime. And then the other way, HISAT achieves faster alignment, but at the expense of some decrease in alignment accuracy, potentially.
Any more questions? How long would it take to align all the short reads from one human genome, one individual, if you use the HISAT-2?
So it's currently the fastest alignment program, and it depends how many reads we have. So I think currently we have hundreds of millions of reads, and then HISAT process like over 100,000 reads. So it sounds like 1,000 seconds, so which is 16 minutes and 40 seconds, I guess. But we have much more coverage now. As sequencing throughput volume have gotten really big, so we expect it might be billions of reads or even more than billions of reads. So the runtime will increase proportionally to the number of reads available.
Okay, thank you, everyone.